In this recording, we're going to discuss the cell cycle. The cell cycle has two main components, interphase and M phase, or the mitosis phase. During interphase, the cell's doing quite a lot of things. It's growing and it's preparing for cell division. We subdivide interphase into three smaller phases, G1, S, and G2. During the G1 phase, or the first gap phase, the cell is doing normal daily metabolic activities, and it's growing a little bit as well. Most of a cell's life cycle is spent in interphase. It's doing its daily metabolic activities. So mitosis, the next phase we'll talk about, is actually a very small portion of the cell's entire life cycle. After the cell grows a little bit and it's doing its daily functions, at some point we enter the S phase. This stands for synthesis, also known as DNA synthesis, or you may have heard it termed DNA replication. During the S phase, we are in fact making new DNA. When we get done with the cell cycle, from start to finish, interphase and mitosis, when we complete mitosis, we will have created two cells. We start with one, we'll end up with two. Each of those cells needs to be identical in every way, shape, and form. So whatever we find in the original cell, we need to also find it in the new cell that we're going to make. So we are going to need to make new DNA to go into our new cell. We're going to need to make new organelles, new proteins, new lipids. All of the things in the original cell need to go in our new cell. At some point, we need to make some of those. Now, during that G2 phase, we're going to grow a little bit more. We're getting larger because we are creating all of these new components that are destined for our new cell. We don't want to run out of room. We also want to make sure that we get large enough to where we can equally split everybody in half. All right. So once interphase is complete, and again, remember that it takes a while, okay, then we will move into the M phase, or the mitosis phase, or cell division. Now, mitosis in and of itself ultimately ends up with cytokinesis as well. We'll talk about the difference between those here in just a second. So mitosis, good old cell division, okay? Well, are we dividing the cell? We're dividing the nucleus, okay? So mitosis occurs when you are replicating the nucleus itself and everything inside that nucleus, also known as your good old DNA. Okay, so remember, during that S phase, we just made some new DNA, and I told you that ultimately that new DNA would end up in our new cell. So that's what mitosis is for. So here we go. We've got four components to mitosis: prophase, metaphase, anaphase, telophase. Okay. There are three very important things that happen during prophase. Okay, so chromatin condenses into chromosomes. Okay, so before we do anything with mitosis, okay, when we are still in interphase, your DNA is shaped like a giant ball of string. It's called chromatin. Okay? If you can try to imagine trying to separate a ball of string equally, and not losing any pieces, not having to cut out knots, things like that. Sounds kind of hard to do. So what we do is we take the ball of string, the chromatin, and we condense it into chromosomes. And this is what a good old chromosome would look like. Okay, so we have chromosome pairs here, because they do come in pairs. Okay. And so that's number one, okay? So I told you we have three things about prophase. Number one is that we condense our chromatin into chromosomes. Number two is the nuclear envelope begins to break down. Okay, so if you remember back to organelles, there is an additional membrane, that nuclear membrane, or that nuclear envelope, that is protecting our DNA. Well, if we would like to separate our DNA or our chromosomes, right, half are going to go into our new cell, half will stay into our old cell, we have to actually get to the chromosomes. We have to get to the DNA. So we're going to have to get rid of that membrane. And last but not least, in prophase, mitotic spindles form. Okay, they're coming from the centrosome, the centrioles. All right, so they're a little hard to see here, but you can see them really well here. These little lines are the mitotic spindles. They are forming during prophase, and they are just starting to attach to chromosomes. So you can see here in our next picture, they have completely formed and they are fully attached to our chromosomes. So you will have noticed that our chromosomes have moved around from prophase into our next phase, which is metaphase. Metaphase is really nice. Everybody likes metaphase because there's only one thing that happens during metaphase, and that is the chromosomes line up in the middle, or we call it the metaphase plate. So you will notice that these chromosomes have moved. All right, they used to be side by side, now they're on top of each other. How did they get that way? These mitotic spindles that had attached were responsible for physically moving these chromosomes and lining them up. Okay. Now, why did we line them up? Because during anaphase, which is our next phase, we're going to pull them apart. We're going to separate them. Okay, so we lined them up, and now we pulled them apart from the center. Okay, so during anaphase, sister chromatids, okay, which is what we call them when they are still connected. Sister chromatids separate and they move towards opposite poles. So everything on the left will go over here to the left, everything on the right will move over here to the right. Okay. Now our last step of mitosis is telophase. And it is basically the opposite or the reverse of prophase. So 3 things happen during telophase. Your chromosomes are going to decondense. Okay, they're going to go back to that chromatin shape. Your nuclear envelopes are going to reform, and your mitotic spindles are going to break down. We don't need them anymore. You will also notice that we are starting to separate here in the middle. We're pinching it in the middle. This little indentation on both sides, that's called a cleavage furrow. Okay, so this is how animal cells separate. They literally just pinch off in the middle. Now, we have finished separating the nuclei. Now we have to separate everything else, and that's what cytokinesis is for. Okay, so cytokinesis, we're going to split the rest of the cytoplasm and everything in it, so the proteins, the organelles. All right, we split everything right down the middle, 50-50, and we get 2 identical daughter cells. Now, this is a very highly regulated process. There are several checkpoints along the way, and you can kind of think of them as stop and go signals, or maybe like a red light and a green light. The most important one happens in the G1. All right, so where are we? Here we go. All right, so we're going through the G1. Boop, we hit our first checkpoint. We're checking for certain things. We're checking to make sure the cell has enough nutrients and growth factors to continue. We're making sure that our DNA has not been damaged. This one's very important. Why? Because if our DNA is damaged, where are we going next? Oh, we're gonna go make some more? Well, probably don't need to make more DNA if the first DNA is damaged. So here's what happens. If we decide that we don't have enough nutrients, we don't have enough growth factors, things like that, we can stop. Okay, we can just hold off, we can just kind of stop right where we are, let the cell catch up, get a hold of some more nutrients, and make some more growth factors, whatever we need. It's cool, it's no big deal. Okay. Once we get those things, once we can pass our checkpoint, we go right along into the S phase. It's no big deal. Okay. However, if we come to this checkpoint, we're like, ooh, you know what? This DNA has been badly damaged, and we can't fix that. Do you think it'd be a good idea to continue through our, no, right? If there's something majorly wrong with this cell, we're not going to want to continue, okay? So kind of keep that in the back of your mind, and we'll talk about what happens if we cannot pass these checkpoints here in just a second. Notice that we do have other checkpoints. We have a G2 checkpoint. We're going to make sure that the cell has gotten large enough to split in half. We're going to check and make sure DNA replication has finished, and we finished it successfully. Everything looks good. And then as we move through mitosis, we have a metaphase checkpoint We're going to make sure that those chromosome spindles have attached and lined up those chromosomes in that nice neat line at the metaphase plate, so everybody will get separated equally. Okay. All right, so I told you keep this in the back of your mind. Cells that cannot pass through checkpoints for whatever reason. If we cannot fix them, if we cannot get them past that checkpoint, they go through a process called apoptosis. Okay, so apoptosis, which is just kind of a fun word to say, but apoptosis, that's what I get for bragging. Apoptosis is programmed cell death, which sounds very ominous, and it kind of is. Okay, so if we go back just one slide and look at our picture. Okay, we said that as we go through G1, when we get to this checkpoint, we have to stop and really have to take note of, can we do this and should we do this? Okay, if we can do this, and even if we just have to pause for another minute or two, get the last few nutrients that we need before we move on, that's fine. We can do that. Okay, but if we get to that checkpoint and something is just terribly wrong, if that DNA is damaged beyond repair, if something else is wrong with this cell beyond repair, we are not going to want to continue to go through this cell cycle, because if we finish mitosis, well, now we have two cells that are grossly damaged, and we're just going to keep on going. What do we call that? Well, we've made a tumor. Okay, so uncontrolled cell division, that's how you get a tumor. So basically, what happens when you have a tumor? You are, see all these little checkpoints? Boop, boop, boop. You're just blowing through them. You are running red lights. Nothing can stop you. Okay, you are not giving your cells the time to check and see if this is a good idea. You are just replicating these cells, cell division left and right, up and down. You are making cell after cell after cell, and they're all bad cells. There's something wrong with all of them. Okay, now you've heard of this. You've heard of tumors. You can have a benign or malignant tumor. If it is a benign tumor, it is encased. Okay, it is encased and it's not going anywhere. It's confined to its original location. These can easily be removed through surgery, things like that. We can just cut that out. Now, if a tumor becomes malignant, notice that it is popped out of its shell and these cells are on the move. Okay, these cancer cells are now on the move. We call this metastasis. So these cells are free to go wherever they want. Normally, and you can see it up here, normally these cells like to travel through the bloodstream, and where does your blood go? Everywhere. So all these cancer cells are free to go wherever they want, and wherever they end up, they can grow and make yourself some new tumors. And so most people know that you can have, I don't know, you can have a liver tumor, and then, 6 months later, things aren't going well. You do a new scan. Well, now you have a brain tumor as well. Okay, so how did it get there? Those cells are traveling through the blood, and they're going wherever they want to. Okay. All right, so that wraps up our lecture on the cell cycle.